Hi, I'm Janine Cody, Director of the Chromosome 18 Clinical Research Center, and I wanted to share a little information with you about our gene dosage map. And so this is the second short video about our gene dosage map. And one of the things I need to point out is why we even have a gene dosage map. And that's because everyone with an 18q deletion and half of the people with an 18p deletion have unique regions of duplication. So that means they have unique genes. So it's not a syndrome in which everyone has the same underlying genetic variation, abnormality, whatever you want to call it. Everybody's unique. So how do we deal with that? And how do we provide families with information that's going to be helpful? The way we do that is, instead of looking at whole syndromes, we look at the genes. So we try to understand the role of each gene and then compile that information into an individualized syndrome description. And in order to do that, we have to know the role of each of the genes. So this is why we have a gene dosage map. And in the first video, which I hope you've seen, I talked about how to create a personalized syndrome description. But that doesn't really cover all aspects of the gene dosage map. And so I wanted to also talk about the other side of the map, or the other aspect of the map that is relevant to the rare related disorders. And so we get questions such as these that we want to understand. We want to be able to help families provide answers. And so for example, my doctor is trying to decide if my child also has disease X, and could it possibly be related to their chromosome 18 abnormality, or is it completely different, unrelated, and it gets treated just like anyone else with condition X. Or we also get questions like, does anyone else with 18p minus have a Y condition? And so even though it might be ultra rare, we want to be able to try to answer that for them. Or also, my doctor can't quite diagnose what's going on with my child. And so what are the other rare diseases that might be associated with chromosome 18 that would have some of the same symptoms that my child is having? This would help my doctor nail down exactly what's going on. So this is why we keep track of the rare, very rare conditions. And so in the gene dosage maps, in the first video I just talked about the clinical map, and that it only has the information for those features found in 2 to 100% of the people. So where the chromosome 18 condition is the primary cause, or the genes of the chromosome 18 conditions are the primary cause of any phenotype, feature, characteristic, whatever you want to call them. And so they're color-coded in the map, like everything I do is always color-coded. And so the pink were those things primarily caused by the chromosome 18 duplication or deletion seen in more than 50% of people with that chromosome 18 change. And the orange are in less than 50%. But the lower end of that is more like, 1 to 4% of people. So they can be very rare. But there are those things that are even more rare. And those are often caused by a couple different mechanisms. And so sometimes they're caused by the chromosome 18 deletion or duplication and one more thing that has to go wrong in order to have that. So they're going to be pretty rare. And sometimes there are those conditions that are caused by the chromosome 18 condition and 2 or more other factors. So I want to talk about that a few minutes because there's so much variability with the chromosome 18 conditions, and this can help explain some of that. So the conditions that are caused by the chromosome 18 deletion or duplication and one other factor we call conditional disease. This is conditional on one more thing. And so those are basically two types, either recessive disease or two gene diseases. And then the other are caused by many other factors are polygenic disease. So let me talk a little bit about those. So recessive conditions, as I hope you know, are caused by a mutation, deletion, non-functional problem with a gene when the gene is not mutated from both parents. Now the parents are carriers. They have no idea they have a recessive, they're carriers for a recessive condition. But when they happen to each donate their chromosome with the mutation on it to a child, then the child has two copies, both copies of their gene being mutated or non-functional, then the child has that condition. That's a recessive condition. But in someone who has a deletion of 50 genes, that's functionally like having a mutation or in any one of those 50 genes. It's not there. It's non-functional. And so you could have, theoretically, a revealed recessive condition in which any of these 50 genes are essentially in carrier status. They're like being a carrier. And if the intact chromosome that person has a mutation in one of those genes, they could then have that recessive condition. So that's a revealed recessive condition. Another example is disorders caused by more than one gene, when two genes have to be non-functional or have a mutation or a certain configuration. And an example of that is facioscapulohumeral dystrophy type 2, FSHD2, which I hope you've heard about before. We've talked about it before. We've had special investigations into this. And it turns out that that can result from a deletion of the SMCHD1 gene on 18p. In addition, in order to have the disease, you also have to have a certain configuration of the DUX4 gene on 4q. So both of those conditions have to be met before you have the disease, at least as far as we know so far. So that's also a conditional. So there's two types of conditional: the revealed recessive or the two different gene, a non-chromosome 18 gene condition. So those are the conditional. Then there's the conditions that result from many other factors, polygenic disease. And you, I'm sure, have heard about the genetics of many of these things. For example, autism, Alzheimer's disease, Parkinson's, many mental health disorders, schizophrenia, for example, are thought to be caused by a multitude of genetic factors that all converge on a single pathway to make something non-functional and result in disease. So in most of these cases, all those factors are not known. But, for example, on chromosome 18, there are a few genes that have been found to be deleted more often in someone with autism than they're found deleted in someone without autism. So that gene is thought to be a risk factor for autism. Well, we keep track of that in our gene dosage map. Not something most families are going to need to worry about or care about, but as interventions and information about autism is learned, it's always good to know which genes may contribute to that. And for people with who have been diagnosed with autism and have a deletion of that gene, as more is learned about the cause of autism, just as one example, we might eventually look for the deletions or mutations in those other genes to understand exactly which type of autism they have. So it's important really for the future, not so much for everyday life for most of us who are concerned about chromosome 18 abnormalities. And the way you get to that information is just like the other gene dosage map. You have to go to our website here, and you go to where it says Gene Dosage Map, shown in the circle. You click on that. It takes you to this page, And the important part of that page is right here in the middle, and it shows our two types of gene dosage maps. In this case, I've already talked about the clinical maps. In this case, these conditions are included in the scientific maps because they have less relevance to the vast majority of people with chromosome 18 abnormalities. So we're interested in the scientific maps, and these maps include information on every single gene and organized by the gene as opposed to by the condition. So here, I'll show you that. Here again are our data shown on the University of California Santa Cruz Genome Browser. Always showing, it's a partial screenshot again, and it shows chromosome 18 ideogram across the top, sort of the pictorial map of the chromosome. In this case, the red box is around just the P arm, and that indicates what's in the bigger box below. And so you can see here down this side are not the condition names like they were in the clinical map, but these are the actual gene names here. And here's the color coding. Now, first thing you need to know is blue means, as far as we know, it's a deletion or duplication. Well, this is the deletion map. So, as far as we know, a deletion of that gene does not cause a problem in anyone with a deletion. So, they're blue. Don't worry about those. The purple are the conditional, and the green are the risk factors. So and again, these are designated by the gene name over here. If you click on the conditional, just like before, you go to a secondary page in the genome browser, and then you click on the gene name, and it takes you to our own website, which shows you the information about how that gene causes disease. Again, this is probably not something you really are going to want to look up. Probably what is going to happen is you would contact us and say, hey, there's this problem, I don't understand why it's happening, no one else that I know with 18P minus has this issue, could it be related or not? And we would look it up. So this is why it's the scientific map. It's less likely to be used by families, but I want you to know it's there. And we have this information, and we keep track of this information. So, in conclusion, the gene dosage maps, to remind you, they're continuously updated. As I said in the previous video, I get daily updates from PubMed on the data released in the last week on every single gene on chromosome 18, every 5 days a week, rain or shine, vacation or no. I get all these downloads. I wake up every morning to about 30 emails that I need to go through every single day. Most have no relevance to having a deletion or duplication, but maybe once or twice a month there's some information I need to add. Doesn't usually change the classification, but often changes the backup information or just to add a reference that might be important later on. And again, this gene dosage map is unique to chromosome 18. No other chromosome has it, so this is why we do what we do, because this is the way to make the chromosome 18 conditions understandable, treatable, predictable. We have a better idea what's going on. It helps us know where to focus for future work. And again, no other chromosome has this. And again, if you have questions, this doesn't make sense, I need to know. That's how we make it better. That's also how we focus our research. We hear questions from you, and that helps us know what's really going on in the real life of people with chromosome 18 abnormalities. So if you need clarification, please email us. It always helps to make the talks better, the website better. The gene dosage map has evolved because of questions you all asked. And I realized, oh, I know this, but you don't know that, so I better explain it better. It's always easy for me to know what's going on, because usually I invented it, I created it, and it came out of discussions with our group, And so, of course, it makes sense to us, but if it doesn't make sense to you, that's all that really matters, and I need to hear your questions. So again, email Annis, and she will get a question to me or to whoever can answer the question. And we keep track of what the questions are to help us make the research more focused on what you need. And so again, if you have any questions, please ask. And anyway, I hope this was helpful. Helps you know a little bit more about what we do and why we do it, and the investment of time that this takes. And I hope it was helpful, and have a fabulous day. Thank you.